So we're going to talk today a little bit about the National Institute of Standards and Technology. I thought, given that we're only 22 miles away, you may not know a lot about what we do, so I'll give you an introduction about NIST and the research being done there. I assume that most of the audience is fairly familiar with microfluidics, so I'll just touch on a few things so that if you're not, you can at least follow where we're going. And then I'm going to talk a little bit about single-cell imaging assays in a project that I did actually about 10 years ago now at the University of Washington, smell in a well. I still think it's one of the highest throughput cell-based assays that's been done. And then I'm going to talk to you about what I'm really working on now and where my project is going for optofluidics and doing flow cytometry in microfluidic systems.
Yes.
So NIST, the National Institute of Standards and Technology, we're actually a government agency under the Department of Commerce, and we are affiliated with maintaining all of the fundamental measurements within the United States of America. So any unit that has to be standardized or calibrated, particularly those associated with trade, NIST is involved and NIST maintains that unit of measurement. But we really have been a part of the United States ideology since the very beginning, where George Washington said that uniformity basically in how we can measure things is very important, and I think that's something that the United States should be focused on. NIST, however, wasn't founded until about 1901, so it took about 120 years for that vision to be realized, but we have a lot of the artifacts that were associated with the United States at the NIST Museum. If you're ever over there, I can show you some of those artifacts. And some of the measurements that you'd be most familiar with, of course, NIST maintains the nation's clock. It's at the university, it's very close to the University of Colorado in Boulder at the NIST Boulder campus. NIST maintains the units of mass and temperature and length, as well as many optical properties. And so we're very broadly distributed into all aspects of science and technology throughout the United States. And so we do that really through three ideas. One of them is creating the fundamental measurement science to develop new measurement technology. One is to disseminate that measurement technology and standards. So creating objects that can be passed around that people can use to validate their measurement in another system. A good example of this is a ruler. You know, you get a ruler, it's got markings on it. How do you know what those markings mean? Well, those rulers, way back early in their creation, were compared against the national ruler, which is in Gaithersburg. It doesn't actually exist anymore. It's now based on the wavelength of light and time. But this is now the goal of measurement science, is to put measurements sort of outside the realm of physical objects, because without that physical artifact, you can't actually easily reproduce that artifact in another location. So now the goal is to link standards to quantum or physical constants of nature. So for example, time is now linked to the frequency vibration of cesium atoms. The length, the meter is linked to the length of time, the amount of time it takes for light in a vacuum to go a certain number of oscillations. And with respect to mass, oops, I can't use this to go back. There's two, mass is still a fundamental object. The kilogram of the United States is buried under 7 layers of very high precision containment under the National Institute of Standards and Technology. It's slowly evaporating. So mass is changing, believe it or not, and that's a problem. It's evaporating at about 1 part per billion every year. That doesn't sound like much, but in the course of the universe, it might not be here. So what they're trying to do is to link mass to a fundamental constant of nature, and they're taking 2 approaches to that. One is what I'm showing you here, which is a sphere of pure silicon. Given the size of that silicon, you know how many atoms are in it, and so therefore it's linked to the number of atoms in a sphere of silicon of a certain size. And the other approach is using an electrical balance, a watt balance-based approach that's linked to the attraction of molecules based on current. So we are a fairly large organization. We have about 2,000 scientists doing everything from smart grid, fire safety, materials, biomaterials, and even cell-based research. We have 2 user facilities, the National Neutron Research Facility and the Center for Nanoscale Science and Technology. We also have 4 Nobel laureates on campus, and I think that we can even claim 6 of them, but they were only there as postdocs, so that's a little bit cheating. These are some of the priority areas, and today I'll touch a little bit on advanced manufacturing and healthcare. Just a little bit about what NIST disseminates into industry. A lot of measurements that go out to industry are actually calibrated at NIST. When you go to the gas station and deliver gas to your car, you expect that it's going to deliver a certain number of gallons based on some standard. Those cans are actually calibrated within each state, but those states calibrate their cans back to NIST. So there's a whole traceability chain of how those measurements go from the National Institute of Standards out into the public. We do all kinds of standard reference materials. We have several thousand products that you can buy, things from standard sand, standard cement, standard oil, standard urine, standard peanut butter. Those are all things that industry uses to calibrate the types of things that they're making in their laboratories, and they need some consistency that they can rely on. And it's up to the National Institute of Standards and Technology to maintain those standards for industry. So we do have growing programs in bioscience and health. There's a lot of work done in health IT. Certainly there's standard reference materials, calibration standards, primarily for biomedical-based imaging. Vaccine quality is now being maintained and worked with through the CDC at NIST. Cell-based assays, part of the projects that I'm involved in, as well as a lot of medical technologies, including studies of bioreactors, biomaterials, therapeutic nanoparticles, and even the American Dental Association has a research facility at NIST where a lot of breakthrough dental composites and things of that nature were developed. So let's get to what I'm here to talk to you about today, which is microfluidics. And microfluidics is the study and control of fluids on the microscale. I like to think of it as using physics and engineering to do interesting things in chemistry and biology, so I'm very much an applications-based person. We say that the flows are laminar, which means that the processes in the flow are dominated by convection, the movement of the fluid, whereas the mixing of fluids occurs over a very long time scale compared to the fluid motion. So things don't mix very easily. And that can be both a blessing and a curse, depending on the particular use that you're interested in. So there are a number of ways to make devices. Traditionally, I think microfluidics came out of the microelectromechanical world, where these were silicon fabrication-based technologies. And that's still, I think, primarily how a lot of the electromechanical work is done in microfluidic device design. Soft lithography, making and molding of microfluidics in materials like polydimethylsiloxane or silicone came out of that technology and is utilized by a lot of labs, especially research labs, because it's easy to make and very much biocompatible, has a lot of nice properties for making things. Embossing is another technology where you can transfer a shape into a material and use that as a microfluidic technology. And then there's also cutting or xerography. People have done cutting in tapes and plastics. We actually did some work in that, and I'll show you a little bit of that today. And then, of course, 3D printing, that's the new exciting world where people are making devices where fluids and interfaces can occur in basically any dimension of the 3 that we can access easily, plus time. So for soft lithography, how this works is we take a transparency and we spin an ultraviolet sensitive photoresist on a piece of silicon. We shine light through the transparency for a negative photoresist like SU8. It hardens that material where the light hits, and then we can cast that with PDMS or some other liquid that we can cure in some way, and we peel that PDMS off and we're left with a replicate device that we can then stick on a surface and create microchannels. In my career in microfluidics, I've been very involved in the use of microvalves to control fluid structure and routing. There are 2 commonly used types of microvalves that are made in PDMS that utilize the elastic property of PDMS to move a membrane open and closed. There's a closed at rest valve, which you can apply negative pressure, pull the membrane down, and fluid will flow across a closed structure. And then there's also a more common, I would think, quake-style valve, as it's often referred to, an open at rest, where you apply pressure and push the fluid, the channel closed against the flow. So as a postdoc back at the University of Washington, I started developing complex microfluidics that could do lots of combinatorial fluid flows and mixing and flow rate control integrated into one system. We actually were developing this to do an olfactory research project that I'll tell you about next, and we never got to using this because I left. But the device has a mixer, which is nice because, you know, like I said, fluids don't mix easily on the microscale, so you actually have to come up with clever ways to mix them. If you just go straight through, those fluids stay very well separated over the centimeters that flow through this device. But if you route the fluids and put them over these bumps which are actually valves so you can turn them on and off, they'll cause the fluid to spin, and you can create, you can spin those interfaces of fluids around each other and thereby reduce the diffusion length that's needed in order to mix the fluids. And so over about 3 centimeters, that should be centimeters, you can get very good fluid mixing. And we utilize these devices to do fluid control in complex multiplexing, where we could pattern and partition fluids, we could create dilutions of fluids, and we could control the flow rates of fluids by integrating a discrete microfluidic resistor network, where here each channel can be diverted into create a higher resistance structure. The flow through microfluidics is the pressure divided by the resistance. So as you increase the resistance, you can slow the flow rate down. It's a very nice way to control flow rate just by integrating these, turning these microfluidic networks on and off. And so this is really nice. We can make very complex microfluidics, but if any of you have tried to develop a device like this in your laboratory with 21 fluidic inlets and over 1,000 integrated valves, this is not easy to use, right? So the expectations are high that we can take complex microfluidics and actually do something useful with them. But the challenges are that it's hard to make these devices. It's hard to reproducibly fabricate them. It's hard to get reliable data out of them. And getting to rapid high content assay, I think, is still a bit of a dream. So how do we, you know, make these systems easier to use and more accessible to general biology? And so we started thinking about how to make systems that you could interface with these microfluidics in a faster and easier way. So we made magnets that you could actually connect fluids to microfluidics just by connecting tubing with a magnet. It would seal against your device. We created manifolds where we have a vacuum network that's distributed around a bunch of inlets that are already in place. So as soon as you set the microfluidic device on top of it, you turn the vacuum on and you can immediately inject fluids. In this case, a 52 inlet device, we can have the whole device loaded in 8 minutes. Whereas if you're doing that, plugging and playing tubing, that can take you several hours, and then you find out 2 of your valves are stuck and you throw the device away. So we also did some work to control fluid flow, and an idea we had was to use pressurized fluid flow. If you're going to be controlling flow to 21 different fluids in a microfluidic device, it's very difficult to try to imagine how to do that with a syringe pump. So we were using a pressurized system, and what we came up with was putting test tubes inside a vial that we could pressurize. But in addition to pressurizing the fluid, in order to account for the change in fluid pressure as the reservoir drained, we actually float the tube inside a larger vessel, and so that meniscus essentially stays balanced. The tube rises as the flow drains, and that keeps the pressure very stable, dependent only on the weight of the tube, essentially. So if you can make very light tubes, you can make very stable pressurized flow rate control. And then again, making these very complex devices, we got frustrated and started to think about, well, how can we just make things simple? What can we do with just a straight channel that might be cut out of a piece of plastic? And so we started cutting plastic layers and tapes and started making devices to do toxicity assays. And I think my colleague Javier Atencia has been here and talked about this before, so I didn't want to go into that. But essentially, we create these channels here underneath this microfluidic structure. We create a chemical gradient over top of the microfluidic structure, put cells in there, and then we can watch the response of the cells to that chemical gradient in that buried channel with time. And this is just, again, a simple device made out of tape. We could do 36-hour toxicity experiments. But when we were making this, we realized, we were starting to align things with folding, and we were doing folding. We thought, hey, we could do a lot more with folding. And so we started making multi-layer devices using folding tapes, making interfaces and connectors. And then we thought, well, another thing that devices with tapes don't have is valves. So how can we integrate microvalves into devices made out of tapes? And so we were able to do that. The real secret to that is finding the right tape, something that adheres well to PDMS. And it turns out that 3M has a particular tape that works great. So if you want to know what that tape is, it's in that paper. And we put microvalves in tape. And then we thought, geez, we're folding, we're doing microvalves. Where did I put my bag? Here. So we decided to go ahead and make a three-dimensional device. And this is what it looks like. This is, I guess, the world's first three-dimensional microfluidic device that has microvalves in the walls. So we kind of scratched our head for a while thinking, what can we really do with this? And we decided we would do a little microcellular organism experiment. So this just shows you that we can route fluids around this cube, and we can have a valve here that turns fluid into this cube on and off. And we decided, well, a colleague of mine was growing these little 1 millimeter long C. elegans worms. So we filled the cube with basically Jello and put the worms in the middle. Turned on a chemoattractant food source over here, and we watched the worms all migrate through the agar over to the source. And then we put in a high salt concentration, which they don't like, and we watch them move away. So we sort of created a lab in a chip. I think it'd be fun to do this with little fish, and then you could really have fish and chips. But we haven't done that. Sorry, bad jokes. Now let's talk about single-cell imaging assays and putting cells in devices to do experiments. We like microfluidics for these kinds of systems because we have dynamic controlled chemical delivery in very small volumes. We can look at single cells or small numbers of cells. And I think an important aspect of this too is that we get very large cell to volume ratios, much like you might find in the body. Things that aren't so easily understood in multi-well plate, where you have milliliters of fluid over a monolayer of culture. And these things are very good for microscopy, so we can do real-time live cell imaging. And of course, because we're flowing through, we can capture the flows and do post-processing of them at a later time. And here again, it's just showing kind of a functioning device, something you might imagine a dynamic multi-well plate assay where you can deliver chemicals in one dimension to do replicate experiments, and then you can switch the flow to another dimension and do an orthogonal treatment. Let me just show that again. So here we have flow going down, then we switch to flow going across. So we might treat a concentration of chemical gradients in one direction, and then we might stain cells to look for responses or metabolites in another dimension. So we had a project back at the University of Washington where we were trying to sense single cells in order to capture the behavior of the olfactory system. And so the background of this project is really, I think, interesting biologically, and it's thought that we can differentiate millions of different smells as well as concentrations of smells, but it turns out our genome only has about 1,000 different receptors for mammals. Humans have maybe 500 functional receptors. And it was discovered, and a Nobel Prize was awarded for this, that each neuron receptor in your nose only expresses one of those genes. And so each cell in your nose has one smell receptor. And so how is it that then we can smell so many different things in this space of combining these smell receptors over a distributed network of single cells? And the idea was developed, well, that there must be some sort of combinatorial code, that different receptors are sensitive to different shapes or functional groups on these molecules. And when you put them all together up in the brain, you get some sense of what that smell is. So how this is done traditionally is you take a single olfactory neuron and you take a pipette that has smell molecules in it, and you take that pipette close to the cell. You puff the molecules around the cell, and you see whether that cell lights up, responds, creates an active potential in response to that smell. Well, whole graduate students' careers were based on measuring the response of 100 cells to maybe a 16-fold different concentration. So we thought there's got to be a better way for people to measure the response of these neurons. And so we developed a project where we would take these neurons and distribute them out one at a time in essentially what's a blueberry on waffle experiment. And we did that in a really high throughput way. So we had an array of these microwells that was about 32,000 individual places for cells to distribute. And we took olfactory neurons from mice and dispersed them in this network, loaded them with a calcium dye which lit up when they depolarize and respond to a particular smell. And then we monitored the fluorescence of those cells as we flow different smell molecules over the array of cells. And so that sounds great too, but there's also a hidden challenge there, and that is that most of the cells in there aren't neurons, and most of the cells in here actually, the non-neurons take up more of this dye and light up than the neurons do. So there's also a challenge in just finding the neurons once you do the experiment. But we found that we could do a double pulse of potassium chloride at the end of an experiment to identify the neurons positively, and then go back and look at which ones responded to the odors previously applied. And so from the 32,000 cells that we had in an array, we were able to positively identify about 1,000 of them. And here you see them responding, poof, one response. This isn't quite as high contrast as I was hoping. Can you see the two puffs of light there? That's when the cells are depolarizing with potassium chloride. So those are essentially all the neurons. We picked them out of the array and quilted them back together, so this only shows the positively ID'd cells. And then we had previously applied odorants, but we went back with the algorithm and identified those cells that had responded to the different odor molecules. And we were able to create sort of a Venn diagram of cells that were sensitive to flour, or what is that, vanilla? Yes, vanilla. Some cells responded to vanilla and flour, but didn't respond to banana or geranium. So we sort of created this combinatorial way to look at, well, essentially 1,000 neurons at a time and their response to odor molecules. Okay, so let's get to the beef of my talk, which is the projects that I'm working on right now. So we have two projects that are in development. One is related to microflow metrology. So NIST is the National Measurement Institute for the United States of America. We're very interested in measurement. And so one of the challenges that's facing the field of microfluidics and consistent continuous manufacturing on the microscale, as well as drug delivery and new bioreactor type systems, is how do we accurately measure fluid flow moving through these microchannels. And we're talking about fluid flow that is about a teaspoon per month, to put that in perspective. Or if you were to think about how long it would take you to drain a 2-liter bottle of pop, it's about 180 years, right? So very slow fluid flow rates. And so it's an open challenge as to how you make measurements of fluid flow within 5% precision on that kind of flow rate. And then the other project I'm developing is actually very closely related. The technology is almost identical, doing optofluidic cytometry, so measuring cells and particles in flow using light. So this is sort of an overview of my project, and we'll start with microfluidics, photonics, and sensing, how we make the devices. And when we say optofluidics, what we mean is photonics and microfluidics come together on a chip. And if you read the literature, that can kind of go one of two ways. What we're talking about is measuring the properties of fluids with optical sensors, but optofluidics is also used to control optofluidics, to control optical properties of materials using dynamic fluids. So using the fluids themselves to create the lasing structures or to change the physical properties of light within microfluidic structures. That's not what we're doing. So they're nice because we can do in situ measurements. At point of delivery, these things can be made small and compact, and theoretically they don't require microscopes. So when I started the project, I joined a team that was trying to do precise measurements of temperature and pressure using optical elements, and they were using essentially three different kinds of structures. This is a micro ring resonator, and this is a photonic crystal cavity. Both of those are waveguides that are essentially resonant to very, very narrow frequencies of light. And there's a similar structure, which is a much larger scale. They are actually integrated into a fiber optic cable. These are called fiber Bragg gratings. And how they work is that you have a refractive index grating that's very fine and very well controlled, embedded in an optical fiber. As you shine light in at a particular frequency or wavelength, those refractive index gratings act like a perfect mirror. And so if you look at what's reflected, there's a very narrow band of that broadband light that comes back, or if you look at what's transmitted, that light is excluded from the transmission. It turns out these refractive index gratings are very sensitive to temperature and strain on the fiber. Anything essentially that changes the spacing between those gratings will shift this wavelength. And so there are very high precision lasers and sensors in use that can trace temperature changes to 0.1 millikelvin. Actually really, really sensitive. These are actually being developed as the next generation temperature standard for the United States. This is what my colleague Zishan Ahmed is working on. But they're also working on integrating these in biomedical devices, putting them on cuffs that can sense blood pressure. As your blood is expanding, the fiber will stretch and relax. And so just simple things. They're being integrated in bridges to detect strain in bridge structures. And so they're being utilized in a lot of different systems. And so we started out putting these in microfluidic structures, just embedding one of them under a microfluidic channel. And we were actually able to show that we could very precisely measure the temperature inside the fluid flow, and we could characterize the shift of the wavelength affiliated with the temperature change within the device. So the idea is you could use these as temperature sensors within microchannels. This was the orientation that we had in this type of device, but going forward, I think it would be useful to think about a configuration where you had much more of the fiber exposed to the fluid. You'd get a much stronger response to temperature. We also utilized these to make essentially a pressure sensor that we then turned into a flow meter. So if you have one of these fibers embedded in a membrane between two crossed fluid flows, you can actually sense differential pressure because the flow that is moving faster will have a higher pressure than the flow that is moving slower. And so that membrane will bend, and that will cause strain on the fiber, and we can actually use that to calibrate this as a flow meter. But because fibers have very high modulus of elasticity, they're not very sensitive to changes in pressure. They're glass. Glass isn't a very responsive material to the types of pressures that you have in microfluidic devices. So you can see here, these are pretty high flow. These are pretty high pressures. The ideal would be to develop something like this that's made out of PDMS itself, thereby reducing the refractive index by several thousand, and getting into pressure sensitivities that are more in line with what you would see in a tissue or even within a cell applied to a surface. So then we decided, well, we should just put light into microfluidic devices by themselves and use that in cytometry applications. And so we started making integrated waveguides, taking optical fiber with light and using microchannels that are filled with high index of refraction material to create total internal reflection. So we can guide light through splits around curves, and we can even focus light through a lens on the end of a microchannel. So here we have the microflow, the liquid, and here we have a waveguide coming through. And here I'm lighting up fluorescene, which fluoresces green when you hit it with 488 nanometer light. And this is just showing that we can optimize the design of the split to get the most light and the most even amount of light through each side. What we're very interested in is measuring fluid flow rate and making those measurements down to the nanoliter per minute flow rate range. Best in the world traceability for measuring flow right now are based on gravimetric standards. This is basically a fancy way of saying a bucket and a stopwatch. So you have a bucket right here, which is a microbalance, and it measures the mass flow from a microchannel over time, and then your time is the other standard that you use to relate to the volumetric or the mass-based flow through the device. These are traceable to the SI unit of measurement, which is important based on mass and time, and also temperature, because that determines the density of the fluid. In order to make very sensitive measurements down to nanoliters per minute, you have to take a lot of care to deal with evaporation. Turns out in a standard laboratory, water from a device like this will evaporate at about 30 nanoliters per minute. So you have to control that, and you also have to control the forces that are associated with the water and the sticking of the water to the glasses and other surfaces as it flows through the system. So it's a very tedious measurement. Most national metrology institutes in the world now, in Europe at least, have these. Most of them are fancier than the one we have at NIST, and most of them cost over $1 million to implement. So they're very big, they're very bulky, and they're very slow. It takes several minutes to make a measurement of nanoliters per minute flow. So that's not how things work in industry. Industry can't wait several minutes to make a measurement in their process. So what's typically used in industry is a heat transfer flow metering method, where you have a heater next to a flowing fluid, and you have two sensors on either side of that heater, and you're basically measuring the convection of the heat created in the fluid from one sense from the heater to the sensor, and you're calibrating that back to something like the gravimetric system. These are great. These are utilized all over in industry, but they're not very accurate. Most of the precision and uncertainties on these are within 5%, and each one only works within about an order of magnitude. So if you want to make a measurement from 10 microliters per minute to 100 microliters per minute, that's 1 meter. If you want to measure from 10 microliters per minute to 1 microliter per minute, that's another meter. So every time you change the range of flow that you're interested in measuring, you have to buy a new meter, and they're about $1,000 a piece. And as you get down to the nanoliter per minute range, the uncertainties at about 100 nanoliters per minute are on the order of 20%. So for measuring the volume of something like a 25 nanoliter microdroplet, it's very inaccurate. Oh, so there are two other common techniques that are used. Particle velocimetry, which is microscopy based. You're measuring particles moving through a fluid, and you do imaging to compute the trajectories of those particles, and from that you can back calculate the flow. But of course the particles can interact with the flow and change the flow, and so you have to be careful about that. And another technique that is almost as good as the gravimetric standard for uncertainty is based on front tracking. That again has typically been image based, but it's a one-off measurement, and it's very complicated by the surface chemistry of the fluid moving through. So essentially how that works is you have an interface, you have your water here, you have air, and you're measuring the movement of that interface with time. But as that interface moves, of course, it's sticking and grabbing along this surface, and so it's kind of jumping, and it's very difficult to control that without very precise surface chemistry on the wall. Best in the world for this is actually in Germany, and their uncertainty is at about 5% for 10 nanoliters per minute, so that's very good. But if you actually think about utilizing this, it has almost no value because you only measure that interface moving through here once, and then your measurement is over, and you've got to rewind everything and start again. And it requires a microscope, and it actually requires several minutes of analysis to make this measurement. So it's not particularly practical, but again, it's traceable to the SI units, and it's fairly precise. So I was sitting in my office one day scratching my head about how we could make, my managers were telling me we needed to implement this interface tracking, and I said, no, this is not going to work, this is dumb. There's got to be a better way. And so I had the idea of, well, what if we were to incorporate a fluorescent molecule, or a molecule that we could turn on with light and measure the displacement of that molecule through the channel. And so that's what we did. We're using a caged fluorescein here. So it's a fluorescein that has a compound bound to it that makes it non-fluorescent until you hit it with ultraviolet light, and then it becomes fluorescible. And so how we do that is we have a waveguide delivering light to a point here that carries ultraviolet light. That ultraviolet light turns that molecule on. That's our start. And then we're measuring the volumetric displacement of the fluid from this point to that point, and then we can determine the flow based on time of flight of that profile. So this was in the NIST News recently, and we've put a patent in for this. This then just shows you the intensity of the light. We actually measure the fluorescence when the light crosses the fluorescent excitation stream here. The fluorescence is captured in another waveguide, and we measure the arrival of that light at this point in a power meter. And then we can determine the time of flight of the wavefront, and we can calibrate that over 3 or 4 orders of magnitude of flow. Now, the problem with that, though, is that the uncertainty of that, the precision with which we know the volume flow through there is dependent on a number of physical challenges. As the flow gets very fast, it turns out our laser isn't powerful enough to activate the molecules or give us enough signal to measure, and so our uncertainty goes up. As we slow the flow down, well, we actually run into challenges with diffusion, but also because the molecules are sitting in the laser path for a longer period of time, we're actually bleaching them and destroying them, so that's taking our signal out too. And really one of the primary challenges is that the uncertainty of the volume in that channel is dependent on the quality of the microfluidic structures that you can make. And it turns out it's quite difficult to measure the uncertainty of a microchannel to less than 5%. And so those errors propagate as you make the channel smaller. Those uncertainties become larger, and they actually dominate the flow below about 100 nanoliters per minute. So with this approach, we can't really get below 100 nanoliters per minute in a dynamic system, but I'm going to tell you that this is no longer true. But unfortunately, we're not far enough along in the disclosure process. I can't tell you about it. I'm sorry, I've only had another few weeks. So we've basically beaten all these limitations, and now we can get uncertainties. We don't even know how good they are yet because we haven't been able to calibrate them on the gravimetric standard, but we predict that we're going to have uncertainties less than 5% at 1 nanoliter per minute. So I actually brought the device. I can pass it around. I prefer you to not open the case, but because the fibers will jump out at you and it's not easy to get them back in there. Yep. So the compound that you fluoresce is inside the fluid that you're flowing? That's right. It's just fluorescein, a commonly used fluorescent molecule. And so these are the devices we make. We just connect optical fibers to them. The microflow channel is right down the middle between the fibers. and laser light comes in one side, and we measure the fluorescence or transmission out the other side. So towards cytometry, you can actually use the same structure to measure microparticles. So here you see a 20 micron bead, which is very much like a cell, coming through, crossing the laser path. And here we're actually just measuring the light that's transmitted through the microchannel, and these are the signals of each of those beads crossing the laser. And we actually measure that in two places. And so we can see the beads crossing at the waveguide position upstream, and we can see them crossing at the waveguide position downstream. And we can see that it's actually very reproducible. Thankfully, these are NIST traceable beads. So NIST has qualified that these beads are very reproducible, and I can say that that's true. They all do seem to have the same size. Waveguide 2 here, you can see, wasn't quite as clean. It's a little bit noisier in operation. But what I'm showing you here are the two trajectories overlaid. You can't see them very well, but there's two peaks here, and the distance between the peaks is essentially the time of flight. So that tells us the velocity of that particle between those 2 points. We've also done some very preliminary tests to look at what sort of resolution we can measure on beads. And I'm going to preface this that these measurements were made with a laser that is 1,000 times dimmer than the one that I have now. And also these beads weren't focused down the middle of the channel, so the beads were distributed across the channel. So the signals weren't very normalized. But again, you can see that the 5, the 10, and the 20 micron beads are all quite distinguishable as they move across the channel. So we eventually hope that we can use this as a standard to also calibrate things for size. And now to talk about bringing it all together for flow cytometry. Hopefully you're aware that flow cytometer is a very important instrument in a clinical laboratory, utilized to analyze all kinds of cells based on antibodies that are targeted to cells for identification of those cells. It's particularly used in cancer identification like leukemia, where they can run and label those cells for cancer biomarkers. They run them through a flow cytometer, and they can measure the number of cells that have those particular cancer markers and the intensity, the number of markers that are on each cell. And very big industry, lots of instruments in use, and the flow rate's actually fairly impressive, 25,000 cells per second. Hard to imagine a laser can analyze 25,000 cells per second, but over the 30 years that these instruments have been in use, they've been able to optimize that to a pretty incredible speed. But there are still, I think, emerging challenges in biology that that's not good enough for. And one of those you might think of would be circulating tumor cells. If you were to try to analyze circulating tumor cells, all of the components of blood, you're looking for about 1 cell per billion. And in order to run 1 billion cells at 25,000 cells per second through a flow cytometer, that takes a day. And no flow cytometer really has the stability to do that. So I think that kind of question is really unachievable. There are also projects being developed where stem cells are being turned into other cell types for manufacturing. You take a stem cell line and you want to develop a liver cell, and you want to validate that that liver cell culture that's going to be implanted into a patient doesn't have any remaining stem cells in it. It's thought that maybe those cells could form tumors once they're put back into a person. So you have to be able to look at millions of cells in order to validate that there are none of those circulating or none of those stem cells residual in that product. In addition, you could think about applying this to look at pathogens in blood or food, where you're looking for bacterial densities of 1 per 10 to the 6th, say, cells in your flow. And you can also use this to set specifications for cell manufacturing. So if you were looking for spike-in control product, for example, where you had 1 cell in 1 million that you could distribute for a lab to validate its count, you need a very specific instrument to create that technology. So you can imagine doing sorting with the device that we've talked about. This is something we're thinking about a few years down the road, but because we can measure time of flight of the particles, we could do computation on that, and then we could apply some sorting parameter to set the cells into a path A or B in a microfluidic system. The nice thing about a microfluidic cytometer too, compared to conventional cytometer, is that your choices aren't really limited to A or B. So in a conventional cytometer, you either go one way or another. But in microfluidics, you can imagine sorting these into 4 or 8 different gradients and setting up a profile of cell responses into different channels. So in order to get to the kinds of questions of 1 in a million or 1 in a billion type analysis in flow cytometry, or even thinking about how do we start to process some of the things that are being done with microdroplets and developing drugs and screening cells in these isolated reactions, we need to think about how we can do more measurements. And I think microfluidics, unlike conventional cytometry, lends itself well to parallelization. And so, you know, if you think you can do one cell in a microflow in one channel, it's not so much of a stretch of the imagination that you could do 8 of them at a time or 16 of them at a time and increase your throughput in that way. So hopefully you've got a sense today that we are using optical materials in microfluidics in order to make interesting measurements at National Institute of Standards and Technology. And we've done that to make what I consider to be, and probably is true, the most accurate dynamic flow meter in the world. And we're starting to use that to address cytometry challenges. We have a proposal in right now. We're in the top 20 for NIST. That's hopefully going to be funded, fingers crossed. They're looking to fund about 3 proposals, and that comes in at about $1 million per year. So we'll have a party. You guys will all be invited if that goes through. Towards flow cytometry, we haven't done anything yet to focus ourselves in the microchannel, but that's fairly straightforward to do with sheath flow. And I talked to Don about maybe utilizing this to look at some of the liposomes that he's making in continuous flow. We want to shrink down the size of the channels and the waveguides so that we can get more precise measurements and finer resolution of cells as they cross through the channels. And of course, flow cytometers now have 6 or 8 colors associated with them, so we'd eventually move to adding more light scattering measurements and fluorescence. But again, that's not particularly difficult. We just add another microchannel around the structure that we can collect light from and getting more power meters. Right now I can only make 2 measurements at a time. So again, this is where we're going. Looking at measuring cells, we can do it now at about 100 cells per second. And I'm pretty confident that we can make measurements of particles well below 5 microns in diameter. We have new lasers in the lab. I just moved into a new lab last month. And so there's a lot of new exciting measurements coming out of our lab. So let me talk to you about opportunities to come work with me at NIST. We're always looking for good postdocs and graduate students, undergraduates, and even high school students at NIST. It's a great opportunity to come and spend a summer or a year working with some of the smartest people in the country. And that's certainly true for the physics lab that I work in. We have 4 Nobel laureates within the physics lab, and a lot of great people walking the halls, so it's really a lot of fun to work at NIST. For postdocs, we have a 2-year federal appointment to come work at NIST. You have to be a United States citizen for that, but you can work in any area of NIST. I hope you choose me. But the deadlines for that application are February and August, and the salary is very, very good. I think actually now it might be more than $68,000 a year. So you must be a U.S. citizen and you have to have a PhD within the last 5 years. We have a graduate student fellowship where we can fund graduate students up to, I think, 5 or 6 years. It's not a problem for graduate students in their second or third year to apply to this program. The deadline for application for that is November 30th. University of Maryland is a partner in that program, so everything is in place for that. For those of you who are interested in that, this is primarily targeted towards women and minorities, but it comes with tuition and stipend and opportunity to freely move back and forth between your campus and NIST. And then we have a summer undergraduate research program. Unfortunately, the deadline for that was 2 days ago. But again, that's open to U.S. citizens and permanent residents. That's an 11-week paid internship at NIST. All the students that I know who have ever done that have had a really wonderful time. It's a lot of fun. You get to see a lot of great research across the campus and interact with undergraduates from all over the country. Hopefully you got a sense of the kinds of projects we're interested in. I have projects at all levels, so postdocs, graduate students, and undergraduates working to develop systems to measure and control microfluidics, particularly in cytometry, basic microfluidic metrology, and even mathematical modeling. Very integrated with a group in applied and computational math, and they've actually done the modeling that has allowed us to improve the flow meter into the very low nanoliter per minute regime. So that's been a very fruitful collaboration. There is a workshop at NIST February 28th and March 1st that is going to talk about all the work that we're doing with the program that we call NIST on a Chip, and that's about creating objects that can be disseminated out from NIST that contain fundamental units of measurement. Most of those are quantum-based, but they thought it'd be important to include microfluidics in there. So microfluidics is a part of that, and hopefully I'll be giving a talk, but I'm not sure yet. The spaces are very limited. Industry is getting at least half of the talks. So if you're interested in coming out to that, I invite you to go to this link or send me an email and I can help you find the right place. And then again, I'd just like to thank all the people who are associated with this work. My postdoc, James Hands. I have a new undergraduate, Steve Meek. Other members of the flow metrology group, my collaborators in the math division, and the work I presented with the olfactory neurons was done at the University of Washington with Albert Folker as the PI. My collaborators developing the photonic chips, Zishan and Nikolai, and then other collaborators we have within the NIST on a Chip program. So with that, thank you for your time. And I'll